Okay, so right, we have seven friends in class here and a couple more online. So welcome, right? So here for our combined science lesson, let's make sure we have the correct one later. You know what? Just pass down the handout first. Please help me to pass it down. Both sides, thank you. Right, we'll be working on two worksheets. So our friends at home, please make sure you get the correct download the worksheets as well. So as we are getting that away, just a little bit of introduction. I believe the previous tutor would have shared a similar set of slides earlier on. Do you all see this just now already? Similar? Yes, yes. Thank you for the nods. Okay. So something a bit special. This is me over here. Right, I've been teaching for about more than 10 years. Okay, and my favorite would be bio lah, right? So bio is my best love, right? Okay, and a little bit about me outside of the classroom. I'm a climber. I go to hiking as well, and I am also an instructor. Right, I'm also an instructor. I teach climbing courses. So if there's something that tickles your fancy, maybe next time I'll see you around in the climbing gym also. Okay, so what we're doing today here, fairly straightforward. Let's dive right in to the worksheet. We should all be looking at this one. Am I correct to say that? Are you all looking at the graph one? Okay, before we even dive into the questions, I would actually like for us to focus on these three topics that I have chosen to put into our first worksheet: movement of substances, biological molecules, and enzymes. Okay, we are looking at these three topics being the first few years. Okay, I want us to think of not which one is the most important, but which one is the most recurring. Which of these topics here is the most recurring, meaning they will keep on appearing over and over again throughout the many future topics as well. I have a suggestion from someone, please. Which topic here is the most recurring? Enzymes. Why do you think so? Firstly, thank you for suggesting. And why do you think so? Raylene, right? I remember your name correctly. Raylene. Okay, Raylene, why do you think enzyme is the most recurring? Oh, okay. Present in almost every reaction, yes, right? So for our topics in bio, chemical reactions wise, only a few topics will have them that we must remember, like respiration, photosynthesis, and digestive system. Right? Okay, but a lot of its focus is only on digestive system. What I am actually thinking of is actually movement of substances. Okay, in your syllabus, Brian, what are the two movements of substances we learned? Osmosis and diffusion. Wonderful. Shooting, which one? Brief definition of one of them, please.
Particles from a region of higher concentration to lower concentration to a concentration gradient.
Down the concentration gradient. But otherwise, wonderful answer. Thank you for correcting yourself and not saying water molecules. Water molecules is for osmosis only. So that is actually a recurring theme that appears in many of our future topics. Almost half of your bio topics will require movement of substances. So get a firm grasp on it, get that out of the way, so that next time in the future topics, you can focus on new stuff. You don't have to keep always, so difficult movement of substances. Okay, so have that first, right? Start off with the correct mindset. Okay, wonderful. Let's dive right in. So here we are looking at raw potatoes. Even before, okay, I'm using the worksheets as a revision for us here because I want to dive into the questions. Okay, so why raw potatoes as opposed to like boiled potatoes, right? So what happens therefore when you boil the potato? How will it affect the process that we are studying over here? Boil in water, there's a lot of water, isn't it? Hold on, hold that thought. So therefore, let me give you 30 seconds to look at the question first and come up with a process in your head. What is this question asking of us? What is this question asking of us? Destiny, do you have a process in your head now? Osmosis. Right? And we are absolutely correct. It is osmosis. Okay. So if you are thinking of osmosis, what does boiling the potato do to the potato that will affect the osmosis things? What will boiling the potato do? Pretty, come. What will boiling the potato do? Don't tell me make it soft and delicious. That's what I'm looking for osmosis. How would boiling increase concentration? Why would water move in? The sound of silence, quite loud. Okay, so Shandell, did I pronounce that right? Okay, Shandell, what does boiling do to my potato? Okay, come, let me throw you a lifeline. Boiling, what does boiling do over here? It's going to damage the cell membrane. Boiling will damage any cell membrane. So how does that affect osmosis? Why it cannot occur?
Because when there's partially
Absolutely, thank you. Osmosis can and must and will only occur across a partially permeable membrane. So if boiling damages the cell membrane either is no longer present or is no longer partially permeable, no osmosis can occur, and my experiment will be invalid. Okay? Right? So we must keep that in mind. We must keep that in mind. So let's get the experiment going. Right? I have 6 of them. I submerge them in different concentrations of sucrose solution. Hey, this is sucrose solution. But Destiny, right? Destiny say osmosis. Why is it not the diffusion of sucrose then? Why do you want to look at osmosis of water and not the diffusion of sucrose? Kathleen. Go on, then cause with sucrose also can what? Right or not? Right? So what we're looking for here is you must remember between sucrose and water, moving across a membrane, sucrose is the one that cannot pass through partially permeable membranes. Okay? So unless the question specifically states the movement of sucrose, most of the time in our questions, we will say that this one is cannot. Ryan, ppm stands for? Wonderful answer, thank you. Okay, so keep that in mind. So yes, we're looking at osmosis of water. Okay, so this is what we see over here. We have a percentage change in mass. What we can see here is we have 2 types of values. The percentage change does not tell me specifically did it become larger or did it become smaller. It just tells me that there is a positive change and a negative change in mass. Okay, Chanel, positive change in mass means that the potato got larger or smaller. Very good. Positive means larger, right? And then conversely, I'm gonna say that negative means smaller. Okay, the potato cells got larger and smaller respectively. Brian, choose one. Tell me, why would the potato cylinders become larger or smaller?
because the water concentration in the potato is less than the solution.
No, choose one first. Which one do you want me to talk about? Which one do you want to talk about first? The positive change one. So the potato became larger, and the reason is because
Water concentration of the potato cell is less than the solution.
so therefore movement of water is from
The solution. into the potato cell.
Very good, except for one rather big mistake. Did we spot that? What is it? It should be potential. We cannot say water concentration. Can? But otherwise, good answer. Thank you, Brian. Right? So over here, we are looking at a water enter cells. And therefore, this one is water exit cells. But for both, I want us to put a big fat carrot over here. Cannot just say water enter, water exit. Because water is constantly entering cells, constantly exiting cells. What would then have to happen for the cell to have a significant increase in size or decrease in size respectively? I'm just looking for one word in front. Do we know what we're looking for? Let's take the orange one for example. When you blow into a balloon, the balloon will just grow in size, correct? You blow 2 more breaths, go larger in size. If you blow 10 breaths, the balloon will then explode. At that point in time, how much air have you blown into the balloon that caused it to explode? How much air would you have blown into the balloon to cause the balloon to explode? Yeah, I'm asking simple question. Please, just think of an answer. Don't just stare at me. How much air would you have had to blow into the balloon that causes the balloon to burst? That's it. Yes, that's it. Too much. Too much. Excess. That's it. Simple concepts. Okay, please try and work with me. Don't just stay here. We're not here to get answers. We're here to learn. Okay? Okay, so only when we have an excess exiting or excess entry of water will we see a significant change in the sizes and the masses. Okay? So we have talked about positive values, larger water, excess water entering, negative values, smaller excess water exiting. There is one set of value that we can talk about as well. What is that? Positive, negative, and neutral. Zero lah. Okay, so therefore, what is happening over here? Why is there therefore no change in the potato cell size? What is the same? What is the thing that is the same? Water potential. At the zero value, where we see no significant change in the potato cell size, this means that the water potential in the cell is the same as the water potential in the sucrose solution. Okay? So therefore, can we say that at this value, there is no movement of water molecules? Brian, no, then yes. So which one is it? So we can say that there's no movement of water molecules? You were correct the first time. We cannot. Molecules, particles are always moving. So instead of saying no movement, this one actually at this value here, no net movement, which is something that Shooting mentioned in your definition for diffusion just now, isn't it? Net movement. What is net movement, Shooting? Net movement means overall movement. So from region A and B, if Shooting, if you give Shandell $10, Shandell, you give her $10, did money move between the two of you? But is there any net movement in the money? No lah, that's it. Okay? So water molecules are always going to be moving. In fact, all particles are always going to be moving. Right? Diffusion and osmosis talks about the net movement, the overall movement. Okay? So no net movement over here. And hence, question is asking us to identify what is the concentration of the potato cell cytoplasm if we take this best fit line kind of thing, we will see that we intercept this no change at approximately 50%. Okay, friends at home, I hope you are following well. As also, if you want to ask questions, just type it out in the chat, and I'll see if I can help you. Okay, so please make sure ask questions if you need to. We are not here to just collect answers. Okay, so in the same idea, the word I want us to look at is remain. Question 2. If the red blood cells remain in their circular and biconcave shape, in 0.2 sucrose. What does it tell me about the red blood cell? It remains the same size and shape in 0.2 sucrose. What information do I have now? In just this one sentence. They remain the same size and shape at 0.2 sucrose. No change in shape and size at 0.2 sucrose solution. What does it tell me about the red blood cell? Yeah, say lah. Hey, you keep it in mind, then I also keep, then we're not here, we just don't do anything. Eh? Any answers? Water, yes, no net movement of water. So therefore, the red blood cell is also at 0.2 sucrose solution. Same concentration there. Okay? Alright. So then if 0.2, it is the same concentration. So therefore this is same concentration over here. Now, if I place it in a 0.15. 0.15 compared to 0.2. Is it more concentrated or dilute? Dilute, thank you. Hey, yes, thank you. I appreciate that. Just say it out loud. We're here to learn, make mistakes together. Okay, so 0.15 is more dilute than the 0.2. So what will happen now when I place the cell in a more dilute solution than before? Again, plasmolyze. Thank you for saying the first wrong answer that we need to look at. Okay, thank you for making a mistake. Plasmolysis and one other word immediately must cancel out. Turgid, why? Yes, plasmolysis, and turgidity are only for plant cells. Okay, so plasmolysis and turgidity are only for plant cells, so we should not even be thinking about them. Okay, so yes, I'm looking at the correct things. Very good. So in a dilute solution, will the 0.15 shooting or the red blood cell have a higher water potential?
Oh,
0.15, very good. So Shendiao, if the 0.15 is more dilute than the red blood cell, who has a higher water potential again? Dilute more water.
yeah, right. Yeah, right.
Yeah, 0.15. So how will water move? Tell me what's the higher and lower now? Into the red cell. So what will happen to the cell now when there's an excess movement of water into the cell? And eventually bursts. Think about the balloon example. You blow one time, never burst. Blow two times, never burst. So it just gets bigger and bigger. The moment you have excess, then you have the burst. So that's lysis. Okay? Right? To lyse is to burst. To crenate is, we must first talk about the shrinking. It will shrink smaller, and crenation is that prickly appearance. Right? Prickly appearance. Okay? Yes, question, Kathleen? You want to ask a question? You don't ask, don't learn, It's on you, You gotta take charge of your learning, Okay, let's go. So here we are still talking about movement of substances. All right, so over here, this is, let me just help you out to understand this better. Starch is inside here. The potassium iodide is outside. So as the diagram suggests, okay? So this one, since primary school, we know already, starch and iodine, you will get that blue-black or the dark blue coloration if they are present together, okay? So therefore, if you look at the next sentence over here, inside the membrane, turned blue-black. Inside was only starch, outside was iodine. This tells me that this occurred. Make sense? Iodine entered. That's why the inside now got starch and iodine, and therefore you have that color change. Did the outside change color as well? Look at the sentence. given to you. The liquid in the beaker remained yellowish brown, meaning only iodine was present. Starch could not exit. So the question is therefore, why iodine can enter, starch cannot exit? Destiny, if you answer for that. Very good, right? Starch molecules are too large. Too large means what? They must be larger than something. Larger than the what on the partially permeable membrane. We don't call them holes. Larger than the through the not holes, not dots, not cuts. What do we call the holes on your face, your skin? Pores. Thank you. Okay, so too large to pass through the pores of the partially permeable membrane. Okay, never see before. What's this? Seen before? Pores.
Yeah, yeah.
Ever? Okay, so therefore C and D are out. Okay, C and D are out. Right? So therefore, between A and B, Raylene, which one would you choose? B is the one that is absolutely incorrect. Osmosis is only for water, and therefore answer is A. Okay, diffusion. I didn't enter by diffusion. Okay, so osmosis, water only. All right, come, food tests. Food tests. A quick look over here. We have already said that potassium iodide is for starch. So therefore, yellowish brown, Shindell, or blue black, which one is a positive test for starch? Yellowish brown or blue black? Blue black. So therefore, let's put a tick over here. We have these two as our positive tests. Kathleen, Benedict's test is for reducing sugars. Very good. Which color is positive?
Great.
Sure. Very good. Okay, so brick grade positive, huh? And last but not least, Brian. What is this mixture? Of all the mixtures in the world, I already narrowed down for you. This is one of your food tests. You only learn 4. Benedict is for reducing, iodine for starch. You're only left with what for protein? What's the name? Burette for protein. And last but not least, ethanol emulsion for fats. Ethanol emulsion test for fats means obviously you have to add. You gotta add fat if you want to test for fat, right? Ethanol emulsion test for fat means you must add ethanol. Are these ethanol? So this has to be Buret's test for proteins. Really, positive test, violet or blue? Violet. With that in mind, let me give you 30 seconds to try and answer question 4. Our friends at home, please feel free to type in chat as well if you want to participate so I can have a look at your thinking as well. Let's hear from Destiny. Question 4, you chose C. What is in egg white? Very good. So egg whites contain proteins, and that's what we're looking at here. So fairly straightforward. C is the correct answer. Okay. If you all have anything they want to ask me to clarify, just do. If not, I'll move on. Okay. unsweetened bread. Okay, this can also apply for just plain white rice. Don't think about brown rice, what kind of rice are, just plain white rice. What does it contain? This experiment that Cheryl is doing will apply for bread, rice, noodles. It is a carbohydrate. In our syllabus, we learn 9 of them. Which one is it? Maltose is sweet. So if I consume maltose now, I will immediately taste the sweetness. This scenario tells me that it is not sweet at the start. Only after chewing for a while, then I will taste the sweetness. What is the major carbohydrate? Yes, starch. Rice, bread, noodles all come from plant material, isn't it? Right? So this one, we're looking at the starting point of starch. Okay, and while you are looking at that, starch, EOY, glucose. Tar. Your display name put tar, so I'm just gonna call you tar. Okay, tar. Starch, not a TY have starch, but don't have glucose. Look at that. Right, glucose is reducing sugar. Here is a negative there, so cannot be have starch. No, cannot be have starch. I hope you've understood that. Okay, so therefore, question 5, we're looking at starting with starch. Why would it taste sweeter? It's actually what Brian mentioned. We are actually producing maltose now. So what caused the breakdown of starch into maltose? Thank you, salivary amylase. Okay, salivary amylase. So keep that in mind. What happened over here? We have starch being broken down by salivary amylase into maltose. Okay, so maltose is the thing that tastes sweet. And therefore, question 5 answer is B. Okay. All right, question 6. Moving on to enzymes already. Okay, fairly straightforward. We're looking for lock and key. Best answer here is what do I say? Okay, maybe one thing I can add on to this diagram over here is this. What do I call this? Oh, I heard the answer. Come share with me, please. Don't you whisper. Come share with me. What was it, y'all? Pretty, come. Oh, Brian, you said it. Thank you. And if this is the active site, this term, this thing, this region belongs to the enzyme. P, therefore has to be an enzyme. Hence, Shooting, the answer for question 6 is very good. I made this question too easy. Make one answer. Okay. 10. Awesome. Let's go. Egg whites one more. So Destiny just now already told us egg whites contain proteins. Okay, proteins. Now, when I say gastric enzyme, means this enzyme is used to functioning in the gastric condition, the stomach. Very good, Ryan. What conditions are we expecting for this enzyme to be working best at in the stomach? Acidity, very good. So we're going to have optimal pH less than 7. Okay, optimal pH of less than 7. All right. Now, the cloudy nature is due to the egg white. So after adding my gastric enzyme, from this cloudy, it turned clear after 8 minutes. Question is asking for more quickly. I want to increase or decrease something. What was the enzyme doing to the egg white? Breaking it down. Simply put, if the cloudiness is due to the egg white, then the absence of the cloudiness is due to the absence of the egg white. So the only reason could be that the enzyme broke down the egg white. Okay? So therefore, for the more quickly thing to be achieved, I need to increase rate of I need to increase the rate of egg white break down by the gastric enzyme? What are the two most fundamental ways for us to change the rate of an enzyme reaction? What are the two ways which we can most easily affect the rate of an enzyme reaction? Temperature, very good, and pH, that's it. Okay, here, 谁叫 temperature or pH. So therefore, if I want to increase the rate of an egg white breakdown by the enzyme, which one should I choose? C, lower the pH of the mixture. Why is it lowering the pH of the mixture? Very good. That's it. You see that? Well done. Okay. Why is D incorrect? I raise temperature. When I raise temperature, my enzymes will have more something. More what? More temperature. The enzymes can have more. Yes, kinetic energy. So then they can move faster, they can react faster. But we do not choose statement D because very good. This if above something. Optimum, thank you. Tells me that you have denaturation. Right, when the enzymes denature, the enzymes lose something. Active site, absolutely. Right? And if active site, look at the previous question, is what is needed for the substrate to bind to. If I lose the active site, there will not be any enzyme substrate interactions really. Okay, no reaction can occur between them. All right, wonderful. Thank you. Let's go. The combination of all those enzyme talk comes to question 8. Can I give you 2 to 3 quick minutes to try question 8 on your own, please? Okay, pretty, come. Share with us your answer, please. Last one. You're absolutely correct. Okay, so we must understand first, right? Why would the seeds even have amylase in the first place? How can we possibly understand for seeds? We have never learned this. Nobody have ever learned that. Nobody ever told you that seeds have amylase. Why do seeds have amylase? What is it for? Go back to the most fundamental meaning of a seed. What's a seed for? Sorry? To grow plant. Very good. To grow plant. Right? So the seed will later on germinate. Right? So as it germinates, it will have its root, then the shoots. During this period of time, can the plant now make its own food yet? No. Then how does it get its food and its energy during this period of time? Primary school we learn, right? Oh, so close. Seed coat protects the seed. The seed leaf. Alright, it's the seed leaf, right? The seed leaf in primary school we say contains stored food for the growing seedling. Now in secondary, we're no longer going to call it stored food. What is this form of stored food in the seed leaf? Starch. Plants store food as starch. Okay, so putting prior knowledge into current knowledge, and it's current knowledge, right? So therefore, the seeds would have to have amylase to break down their own starch stores. Then later on, we will release it as glucose to release energy. Okay? Can All right, awesome. So yes, answer for 8 is D. Well done. Okay, so this one, hot springs, I give you average temperature of 120. Question is, what kind of optimum temperature would its enzymes have? Would its enzymes have? Pretty straightforward question. Shooting, your answer is optimum temperature would likely be where the organism is. Any enzyme in our body will have an optimum temperature near to our body temperature. So if this organism is found in a place that is at 120, it must be able to survive the 120, right? So the enzymes in its body or cells or whatever will have an optimum temperature close to 120. Do we have 120 in our x-axis here? So have we reached the optimum temperature in this graph? And hence, shooting the answer has to be D. Why? Yeah, I explained everything. You just need to tell me answer only. Okay? So it will continue all the way up until we reach 120. Okay? Last question. Trypsin is an enzyme found in our body as well. So that's why we can digest proteins in the milk. Right? Here we are looking at effect of temperature. Effect of temperature. Let me draw two graphs for you. Which one is the effect of temperature? Which one is the effect of pH on enzyme activity? Which one? Shindiao, can you help me with this? Which color is for temperature? Blue one. And the green one tells me the effect of pH. Wonderful. Okay, so this tells me that both graphs have this thing in common. What is this? Optimum. Thank you. Right? It could be optimum temp, it could be optimum pH. Both are true. Okay. So optimum means destiny, for example, if I'm looking at optimum temperature, this means that the enzyme is working at its what? Maximum activity, maximum rate, correct? So if I'm working at my maximum rate, Brian, am I working very quickly or slowly? If I'm working very quickly, do I need a lot or very little time? Very little time. Okay, use that and find the answer, please. So your question is to what's the difference between what and what? Oh, okay. So the graphs I need to be able to identify about the optimum. pH graph is symmetrical. Agree? So that is one thing you need to be able to see immediately. Oh, find your pH first. Always find your pH first. Once it's symmetrical, pH curve. Once it is asymmetrical, it's going to be the temperature one. Okay? For both, this side of the graph, what happens to the enzyme? Denature. The difference lies in the below. For pH, it is still denature. However, for temperature, it's not denature. What is it? Wonderful, really. Okay, inactive. Okay, so that's the difference, Chloe. I hope that rings a bell for you. And hence, Destiny, you chose. The last one is the answer that I hope you will choose for the incorrect one. You need to be able, that's why I asked Brian a question just now. The greater your rate of reaction, less time you take. So I'm looking for a flip. The answer is C. Answer is C. Okay? That's why I put it there. Right? You need to be able to see the less time you take, the faster you are working. Oh. Oh. Okay? Hannah? Yes. Our answer is C. Okay, moving on to our free response questions already. We're gonna move a little bit more quickly here. So for question one, for similar as the first two questions we did in our MCQ here, we must first identify what process are we looking at in question one, part A. Process are we looking at in this question here? Osmosis, wonderful answer, right? Thank you. Okay, so looking at osmosis because, I mean, distilled water only has water. So it has to be about osmosis, isn't it? Wonderful answer, Brian. So looking at distilled water, question is asking us about the red blood cell here. So what words do we use to describe this red blood cell now? I'm looking for two descriptors. Burst is one. But before bursting, it doesn't immediately burst, right? We will first. Thank you for saying that wrong answer. We want swell because expansion is for when you gain heat and expand, correct? Not? You gain heat and expand. We're not talking about heat here. So I want to separate these two terms here. Okay? Because in our minds, expand, yeah, get larger what? I want to really focus on what is actually happening. We're not looking for expansion, we're looking for swell. Well done, Shiting. Okay, so looking at swell and bursts. What causes this to happen? Are we looking at excess entry or excess exit? Excess entry. So what does it tell me about the water potentials? Where higher? Distilled water, thank you. Don't outside, inside. So distilled water, higher water potential. The first thing, whenever we answer movement of substances questions, is to recall my definition. In all movement of substances questions, 80% of your answer will come from your definition. Where will the 20% come from? Where will the 20% come from? 80% of your explanation will be adapted from your definition. The net blah, 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 blah. That 20% will come from answering the question. Question not asking you about osmosis. Question is asking you about what happened to the cell. So after you talk about osmosis, you tell me then why the cell like that. Okay? So we'll start off with the osmosis portion first. We will state who has a higher water potential. Fill in the blank for me, please, Pretty. Very good. Ryan, fill in the blank for me, please.
Question. Oh, did
Same thing we talked about just now, right? Do we remember? Concentration is only for diffusion. Potential. Okay, please remember that. Water potential. Celine, yes, you may. And destiny across the. I want to bring it even further, not just partially permeable membrane, partially permeable cell membrane. Okay, answer the question. And after all this, please remember to say these last two words. Then the marker will be like, you talk so much, you didn't tell me your process. Okay? Now we are done with the osmosis portion. Okay, I tell you water go from where to where already. This is your 80% dimension. Right? The 20% left is to answer the question. Question asks, hey, what happened to my red blood cell? We need to use words that we identify early on. What are the Shenya, what are the two descriptors we are going to use to describe this red blood cell? Swell and burst. Thank you. Right? Do we say, oh, water enter the cell swells and bursts? Is it just water enter cell will swell and burst really? Excess entry. Okay, so you must also out that there's too much water entering. Okay. Right, and something I always like to remind everyone is after copying down something, whether is it from the screen or just listening to your teacher, read your own work first. Right, it's a different thing copying and a different thing reading your own work. So take a minute, read your own words. Make sure you truly and fully understand what you've just written, not just for the sake of copying. Always remember what's the 80%, what's the 20%. Okay, so now conduct another experiment that's similar, but instead of distilled water, I put it into concentrated sucrose solution. Okay, just now the red blood cell was it swelled and burst. Now what happened? Very good, but that is the second word. So just now swell, then burst, shrink, then crenate. Okay. What we're looking for here is the question is asking us about the plant cell. However, no matter what cell you are, the movement of water, the direction of movement of water will be the same. If it entered the cell over here, it will also enter this cell over here. So hence, this one quite obviously is the opposite. This one exited. The thing we're going to draw is also an exit. Okay? But I put this out over here. I want to save time. I'm going to draw. You're going to tell me what is correct or wrong. Okay? Okay. No shame in saying that this is something you would definitely have drawn or would have drawn for this question. Okay? But there is one error here. What is the error? Very good. The cell wall is missing. We must draw this. I'm going to use dark blue for a slight difference here. Because when I look at both cells side by side like this, I need to understand that my cell wall is a layer. My cell wall is a layer. The cell membrane is a line. Because if you look at this printed one for us, okay, eyes on screen, please. If I draw cell wall, this is my cell wall. If I draw cell membrane, the cell membrane is here. So therefore, obviously, we're not going to draw two lines overlapping, so that we always draw two lines only. Okay? So therefore, when this happens, this is very important to show that the cell membrane has pulled away from the cell wall. You need to show this. Okay, you need to show that you know that the cell wall is a layer or the cell membrane is a line in our drawings. Okay? Come, go ahead. Draw this out now, please. And after you're done, then I'll ask you to give me two descriptors as well. 就听选掉 that your like school study notes or something? Is that your school study notes or something? Or is it your make one? Yeah. It's like they condense everything into a worksheet. Come, I have two descriptors that we need for this cell here. We do not necessarily need to know the word for this state, which is plasmolysis. But we need to, at the very least, be able to describe plasmolysis. There are two things that we need to talk about, one of which I have just mentioned. No, plasmolysis is the state now. There are two things you need to say to describe plasmolysis. Flaccid is about the layer of cells becoming flaccid due to the plasmolyzed cells. Can see the difference, So this thing, what is this? I did this just now, I said something. Oh? What thing separates? What pulls apart?
Cell.
Yes. Yes, very good. So therefore, cell membrane, oop, too many lines. Cell membrane pulls away from the cell wall. Secondly, look at the two diagrams. Identify the organelles inside. These two things are the nuclei, so that's the nucleus. No change in nucleus. What about this two? That vacuole, what happened to it? Yeah, absolutely. Please spell the vacuole correctly. Vacuole. Okay, so the vacuole shrinks and the cell membrane pulls away from the cell wall. That is plasmolysis. Okay, so if you look at the next page, it's just asking you to explain that. Like I said, 80-20, 80% from osmosis, 20 from answering the question. Question asking you about appearance. So in a similar fashion, we're not gonna spend time doing it here because there's a reason why I work this out with you. Okay, if you very quickly, eyes on screen, lend me your ears. The answer will be concentrated sucrose solution has a lower water potential than the cell's cytoplasm, causing excess water to exit the cell. All the same. Then you answer the question. The exit of excess water causes the cell to done. Okay, right? So Movement of substances can be very easy. Yes, it can be a little bit tedious, I admit to that, but the points are there. 80% definition, 20% answer the question, 100% marks yours. Okay, let's move on. Question 2. We are looking at oil stains. Which biological molecule family does oil belong to? Again, lipase is an enzyme, which are... No, are you telling me that oil contains lipase? Did I understand you correctly? No, oil contains... Pretty? Yes, fats. In a way, oil is liquid fat. Okay, oil is liquid fat. What is solid fat? Butter. Yes, butter. Then buttermilk, then oil. Okay. All right. So they were washed with biological washing powder. What's so different, what's so special about biological washing powder? And then when I add biological washing powder, the oil can disappear. Okay. I washed it at different temperatures. So this is a clue for you. This thing that causes it to have this biological name is affected by temperature. What topic are we at now? Enzymes, absolutely enzymes. Okay, so biological tells me contains enzymes. Okay, so 15, 40, 75. How do we choose which temperature will result in the oil stain becoming the faintest? At which temperature will the enzyme cause the oil stain to be the faintest? The temperature that's closest to the enzyme's what? Optimum temperature. If the question never state, we will take the optimum temperature to be closest to body temp. Our body temp is 37, and hence here we're looking for 40. Okay, very straightforward. First one, 40. How to find the next two? We have 15 and 75. One is no change, one is little bit of change. How do you choose that? Think back to just now, if you all drew with me the temperature and pH graph in the MCQ. What is the difference? What is the difference when temperature is above, when temperature is below optimal? Do you think? 50 is inactive. So what does it mean about the enzyme? What does it mean when the enzyme is inactive? Never work at all. Then what about denature? Over. Okay, this is interesting here. Overwork. Okay, so another perspective that I can share here is denature, D for denature, D for die. Enzyme die doesn't work at all. Okay, inactive is like you are asleep. You are very sleepy. You cannot asleep, you are sleepy. But you can still do some work very slowly. Hence, this is 75. The enzymes are not doing anything at all, so no change in oil state. The sleepy, inactive enzymes are doing a little bit of work at 15. Okay, so we are 40, 75, and 15. So question is asking us to identify what removed the oil stain. Really? B part 1 then is lipase. Okay, so this is lipase. So do we agree with Eunice's statement? We agree with Eunice's statement here? Pretty? We say no because... Yeah, it's that whole denature thing. Right? So denature, we got point from this one. Right? So at 90, this one, you will denature. When you denature, enzymes lose something very important to them. Very good. Lose the active site. Cannot do what? Without the active site. Okay. Catalyze the digestion of fat and oil stain. Can I? So with that in mind, we know that the biological washing powder contains our enzymes already. Part C is asking for the test for it. Okay. Kathleen suggests ethanol emulsion test. Chanel, do you agree with her? I don't say wrong. Not yet. Chanel, what do you think? Are we looking at the ethanol emulsion test here? Why not? Ethanol emulsion test, not test for ethanol. Ethanol emulsion test is test for what? Yes. Chanel, we talked about it just now. Come, Raylene. What is the test that you think it should be? Why Burex? Why test for protein? Hey, read the question. Yes, read the question. In B part 1, what did we write over here? Lipase. Lipase is an enzyme, and all enzymes are proteins. Thank you. So this one, I got 90% success in tricking your I did this many times already. So people happily, oh yeah, fat, blah, 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 come to the test, ethanol emulsion. Okay? All right, so please keep that in mind. The test we are going for is the Buret's test. Okay, we must first state Buret's test for proteins as lipase is an enzyme that is a protein. Then we go on to describe the Buret's test. You can go for the sodium hydroxide and copper 2 sulfate thing, or you can just straight away add Buret's reagent. Okay, right? So this one very straightforward. This one is a copy-paste kind of thing. You all can go back and check your notes and stuff. The answer key will be provided once all the sessions are done. But one thing to keep in mind is these food tests can only be done on aqueous samples. If I give you a chunk of chicken cubes, you cannot do the Buret's test. What must you do to the chicken cubes first? Melt. It will burn before it melts. You just mash it and you pour water. Then you filter. And you will only test the water portion. Okay? Yes, you have to mash it first. Okay? All right, so let's move on. All right, so here, once more, starch and saliva. Starch and saliva. What is the enzyme we are interested in here? Salivary amylase, thank you. So let's hop right into the table and understand the differences. Between flasks Q and P, the difference we're looking at here is 5 and 2 cm3 of saliva. What factor are we studying? Very good. Concentration. So this is enzyme. The effect of enzyme concentration on the rate of a reaction. Pretty, what does this do? When you boil something, we boil an enzyme. Yes. You denature the enzyme. 诗婷, what are we studying for flask S? Look like same as Q, huh? What's the difference?
But one is
Is there something else that we can look at? Right? Okay. So if boiled saliva, we say denature. Here, we will say what happened to the enzyme. So what you very good, inactive. Okay. Unfortunately, I forgot to take away the numbers in your paper. So the curves are there. Right? The answers are there. Okay. So simply put, flask R, we have curve 4 because why? In flask R, denatured meaning the amylase will not be able to break down the starch at all, and therefore 100% starch remains. Make sense? Okay, right. 1 and 2 will have no starch by the end because in 1 and 2, the amylase is functioning properly. Flask P has a higher concentration of the enzymes, therefore it reaches 0 faster. So therefore that is flask 1, curve 1. Okay, very straightforward. The last question, very simple. Looks like an entire paragraph here. You actually only need to zoom into one word for you to find the answer. Can I have you choose that one word as the answer, please? Choose it. Destiny, what word do you think we should zoom into? Acidified. Why do we want to look at acidified also? Very good. What would that do to my enzyme? Denature. And we also see it over here. Alright, so therefore, question is asking for which one will result in the exact same curve. Our answer, Ryan, should be curve. Fortunately, that is the correct answer. Okay, so it's curve 4 as well. Right? So explanation here, very straightforward. You're just gonna tell me that hey, acidifying it or adding hydrochloric acid is gonna denature the enzyme just like how boiling does. Now, there's a very interesting twist to this question because question is asking you to explain why it exactly matched. Question is not asking you to explain why no digestion. Do you understand the difference between these two answers? We are not asking you to explain why no digestion. We're asking you to explain why the same as flask R. Okay, so points that we must have over here, points that we must have. Okay, acid denatures amylase, which is the same as boiling denatures amylase. Okay, so this is one key point that I think it will be very easily missed out. So let's jot this down and be done with the first 10. Yeah, estimate enough. Oh man, short by one. Here we go. Back down. All right, ladies and gentlemen, let's move on to the second handout. Okay, so as you can see, we are really pressed for time. So we're gonna move on a little bit more quickly. I appreciate your prompt answers. As long as you have them in your head, you can share them with me as well. Let's go. Nutrition, transport, and respiration. This is an excellent opportunity for me to remind you again. What is the most recurring topic we're talking about just now? Movement of substances, because each of these three topics involve movement of substances somehow. Okay, you have absorption of nutrients in nutrition in humans. You have exchange of gases and nutrients for transport. You have exchange of gases in respiration. Okay, very important. So let's dive right in. We have naming. Whenever we see this word hepatic, I want us to remember the liver. In a human body, we have words that makes our life easier by grouping things together. What I mean by whenever we see the word hepatic, it's going to be associated, it's going to be connected, it's going to involve the liver somehow. Okay, next up is artery. Artery, A for artery, A for away from the heart. Okay, so arteries always transport blood away from the heart. Its counterpart, the vein, brings blood into the heart. Let me see that over there. Okay, so artery away, vein goes in. We learned 3 blood vessels though. Artery, vein, and capillaries. So I'll share with you right now. Arteries and veins are transport vessels, meaning they transport blood. That's all. Nothing else happens. Capillaries are not transport vessels. Oh, how do we define the function of capillary? They're not for transport, so we cannot use the away or towards. What happens at the capillaries? Absolutely exchange. Okay? Exchange. Exchange of thermal materials occur here. Good try, Lucas. We're looking for exchange, Okay, so with that in mind, blood vessels always have this two name series. Your first name, your last name. First name is always the organ you're connected to, hepatic. Last name is direction of blood flow, artery or vein. That's it. With that in mind, we have to identify E and F. Really, choose one and identify them for me, please. Is the wonderful. Now, the hepatic portal vein is a very special vein because it is the only vein that does not bring blood from an organ straight to the heart. It goes through another organ, and as you can see in this diagram, it goes to the liver. Okay, so this brings us to our last organ, vessel over here. Shadel, can I have the name of vessel E, please? Chanel, use this. Artery away, vein towards. So therefore, yes, hepatic vein, very good. Hepatic vein. Okay. Your option B, is it the same as option B? Okay, let me change, please. It should be F, then E. Because if not, then no correct answer. Typo on my end. Can Okay, so it has to be B over here because what is this wormy boy over here? What is that? Well done, Brian. That is the small intestine. And what happens at the small intestine that is relevant for this question over here? Excellent. Absorption of glucose. So once glucose is absorbed from the small intestine into the bloodstream, it will first be brought through the hepatic portal vein to the liver. The liver is like a bank. When you carry too much money around your pocket, you go to the bank, you store it there. The next day, your pocket all empty. You go to the bank, you take money out. So when blood has too much glucose, the liver is the first stop. Hey, you got too much, let me store some for you. So that in the future, if you encounter a scenario where there is not enough glucose in the bloodstream, the liver will take those stored, break it down, and return it back to you into the bloodstream. Okay? All right, so I'll say it's B, very straightforward. Move on. The reason I put this question over here is to share with you that when we see weird words or things that are so big you cannot even pronounce, I want you to just ignore that word and look at the information that comes after. Kathy, you want to try and pronounce that word? Really, you want to try?
Cholecystectomy.
Very close. Cholecystectomy, right? A cholecystectomy is, simply put, once you arrive at these four words, that's it. It's not application question anymore. Ignore the cholecystectomy. Okay, right? So there's just I put it in there for fun. It's one of the episodes of Bio Tongue Twisters. Cholecystectomy. Very good for passwords as well. Okay, right. So removal of the gallbladder. Whenever we talk about removal of this, removal of that, the first thing that comes to mind has to be, ooh, no. What do you do? Gallbladder does what? Gallbladder does what? Store bile. And I'm glad you say store bile, but I want to add in one word here. Store then keep forever. So the gallbladder also. So why do you not say produce bile? Yes, yes, yes. Because bile is by the liver. Alright, please keep this in mind. Do not make this mistake. Oh. What are there more white blood cells after a person undergoes a surgery? Surgery means I must cut you open, right? That's like a wound, correct? So that can expose you to infections, right? No matter how big or how small, the body is very sensitive. The moment there are infections, white blood cells will increase in concentration in your bloodstream to patrol, to clean up, to remove those pathogens. Okay? Good question, Chloe. I hope that quick answer answers you. All right, so storage unit is bound. Now that we understood what's the function of the gallbladder, if we remove the gallbladder, this means that this function is gone in green. Is anything in blue affected? No, So therefore, you look at statement 3. It says less bile is produced. Is that a correct sentence? Incorrect. Okay, so we still have bile there. All right. Let's head up then. Decreased glycerol secretion. What is glycerol? What is glycerol? Glycerol is part of a fat molecule, right? Glycerol is part of a fat molecule. So therefore, this one, do we secrete glycerol? We don't produce glycerol. Glycerol comes from the breakdown of fat. So since this has nothing to do with it, that is not our answer. Okay? Anything to do with amino acids? No, What? It's the function of bile. What is the function of bile? What? Do you have an answer? Function of bile? Emulsify fat. What is emulsification? Very good. So, with enough bile, I will be able to break up those large fat droplets into smaller ones, increase surface area, higher rate of fat digestion. But now, without the gallbladder to regulate the release of bile, the liver will just keep producing. Have fat in the small intestine, also release. Don't have fat, also release. Meaning, bile will not be as efficient anymore. And hence, fat digestion occur at a lower rate. Okay? Alright, so answer here is D. Okay, let's very quickly identify these structures that we see in both their bloodstreams. This one is red blood cell. These are, what are these? They're much larger than red blood cells. They have these black dots inside of them. Yeah, the white blood cells. White blood cells are larger than red blood cells. And number two, white blood cells have nuclei. So that the black stuff are the nuclei. Okay? And finally, what are these dots? What are these dots? Platelets. Think about the functions of these three components of blood. And therefore, Sarah's blood can or has what compared to John's? Destiny. What do you think is the answer here? Sorry? D. Yes, Sarah's blood can transport more oxygen because Sarah's blood has more red blood cells. Okay, has more red blood cells. Wonderful. Question 3. Very straightforward. Let's go. Oh, not ABCD, W, X, Y, Z. But that doesn't affect question. Okay. Okay. So, vena cava. What is the vena cava? It's a vein. Very good. So, as I said earlier on, vein goes in to heart, right? Veins bring blood in to the heart. So if you look at direction of blood flow, it has to be towards, right? Okay. And moving on from the word vein, we can look at the muscle layer and lumen diameter. Veins have thin or thick vessel walls. Veins have thick or thin? Thin, very good. Why? Why thin? And in contrast, why do arteries need thicker muscular walls? Arteries are blood vessels. Vessels transport only. Yes, what's the word there? Thank you. Withstand. The artery transfers blood at high pressure. So the wall must be thick to withstand that high blood pressure. Okay, so this one, arteries, high blood pressure. Okay. All right. So with that in mind, definitely has to be blood vessel Z. Okay. So remind yourselves properties of veins, thin walls, wide lumen. Okay. Because the blood pressure in veins are very low, we want to make it as easy as possible for blood to flow already. So we want to widen the lumen. Okay. Not transported by blood plasma. Not transported by blood plasma. Pretty, can I hear from you, please? Hey, very simple. Don't choose the thing you don't know. Don't claim, oh, this one never hit before, should be answer. Don't choose the thing you don't know. Shiting, do you have an answer for me for question 5? B. Breathing through this oxygen. You are correct. Question is plasma. What transports oxygen? Red blood cell, ah. So the oxygen is not on the red blood cell. It is in the red blood cell and therefore cannot be in plasma. Make sense? All right, read the question properly. Oxygen is transported in the red blood cells and not in blood plasma. Okay? If I took away the word plasma, then oxygen is transported in blood. But I specify plasma, so it's not oxygen. Okay? The other three things are transported by plasma because they are dissolved in the blood plasma. Okay? Fibrinogen is a clotting protein that is not required of you in your combined science syllabus. You just say clotting protein for clotting of blood. You need me? You recognize fibrinogen? Oh, good on you then. Okay. Testosterone. Why? Oh, okay. Right. Testosterone is a male sex hormone. Okay. You only need to know of like hormones next time. Yeah, maybe yes. Okay. Urea is also transported in the blood plasma as well. Okay. Awesome. Let's go. Direction of blood flow. Direction of blood flow. Think about what I shared with you earlier on. A for away, a artery away. So all the arrows pointing away are arteries. So they gave you already blood vessel 1 is the aorta. This is the aorta. Okay, actually the entire thing is aorta. What are the other vessels? Can make use of question 4 as well. Question 4 gives us answer for vena cava already. Really? Can you show me the answer for question 6? Chose C for the correct answer. Very good. Okay, so we are looking at word associations. Hepatic, just now we said liver already. We are looking at a few other words over here. We have renal. What organ do we associate with renal? Kidney, thank you. What organ do we associate with pulmonary? Lungs. Brian? Coronary is for heart. Okay, right? Because blood flows through the insides of the heart too quickly for any exchange of nutrients to occur. So therefore, it will exit together with all of the other bloods via the aorta and then has its own branch to supply oxygenated blood to the muscles from the outside, not from the inside. Okay? Okay, fairly straightforward. So again, revising pulmonary and vein. This one, Destiny, the answer should be pulmonary means lung. Vein means go into the heart. C, right? Because if it goes into the heart, means it came from the lung. Just absorb lots of oxygen. Answer is C. Okay. Lactic acid. What process comes to mind when we think of lactic acid? Excellent, really. And this question is actually more about understanding the graph than the content of anaerobic respiration. Let's look at statement one. There is a maximum level of lactic acid that can be produced by the body. We cannot actually tell that. Why not? For us to be able to see a maximum concentration or maximum quantity of anything, the graph should show me a plateau that we have no longer any more increase throughout my entire experiment. What this graph shows me instead is that it peaks at this point, but this peak may not be its maximum. Okay, so maximum, we are looking for plateaus. No lactic acid in the body. Absence of physical exertion is incorrect because at time zero, where the person has not started exercising yet, the graph tells me that there still is some lactic acid. Okay? Part C is the only one I would say requires content, which we have already stated over here. Really reminded us the lactic acid is produced via anaerobic respiration. And it's not going to be about aerobic. And hence our answer is D, because if I produce more quickly than I remove it, it will start to accumulate. And that is what we see over here for question A. Okay, so question 9 over here, we're looking at rate of oxygen uptake. This is when the person stopped exercising. I want you to think back to the last time you had your 2.4 or 0.6. 2.4 already, right? Okay, when you pass the finish line, from your breath being, does your breath really go to? Doesn't, right? Okay. Hopefully, it doesn't increase like crazy. You stop exercising already, suddenly increase, and go hospital already, this one. Okay? So definitely not A. There are two other lines that also definitely wrong. Which one? Which two lines definitely wrong? B and D, because they go to 0. That'll be our last 2.4 ever. Okay? And hence, you're going down to C. Why does it not just immediately drop to normal? You stop running already, what? You don't need the additional oxygen, right? Shendel, why does why does rate of oxygen outtake not immediately return to normal levels? Why? Breathing rate increase, rate of oxygen uptake increase because you exercise. You need the additional oxygen. You stop exercising, you don't need the additional oxygen for that energy release anymore. So why does it remain high for a short period of time right after? Why does the graph do this slowly go back to normal and not this go back to normal? Celine, yes, correct. Found it. That's absolutely wrong. Come, anyone? To repay the oxygen debt. Yes. Does it ring a bell? Repay the oxygen debt. These are concepts that we are learning, Shindell, what common sense? No, common sense is, yeah, okay, because you have encountered it a lot of times. But if you're not able to explain why is that common sense, then it's not it. Okay? All right? So yes, we're trying to repay the oxygen debt because of the harmful lactic acid that we produced earlier on. We need that additional oxygen to break it down. Okay? We need that additional oxygen to break it down. All right, last question. I would say this one is the most straightforward that I made. Okay, it's the most straightforward one that I made. Identifying correctly the structures of the alveolus and being able to tell me what its function is. Okay. It says it's B because why? Okay, the answer is B. It's very important for the gases to dissolve first because we must understand the blood does not transport gases if they are gaseous states. There are no gases in the gaseous states in the body. Everything is aqueous. Okay, so when we say transport oxygen, transport carbon dioxide in other forms, not in the gaseous states. There should not be air bubbles in our bloodstream. Because you all see all those medical drama hospital, everything, you see the doctor take out the needle, oh, like that. You're not doing this to look cool for fun. They need to remove the air bubbles. Because if air bubbles are injected into your bloodstream directly, air bubbles are small, yes, but you have even smaller blood vessels. If the air bubble were to travel to a small blood vessel and gets stuck there, it could create this airlock that blood cannot flow through. And then that organ will be stuffed of oxygenated blood, eventually die. Just one tiny air bubble can do that. All right, so you gotta be careful about that. So no gases in the gaseous states. This dissolving is very important. Okay, awesome. Let's go. So of the three questions that come next, I want us to give a quick glance of the three. Let's choose one. We'll look at it together, class. Yeah, because at the end of the day, I'll pass you the answer key anyway. So let's look for the one that we want to work on together. Oh, it's liver. Which one? Which one? You got learn about the heart, right? Heart structure, have, right? Son. Very good. Thank you for pointing that out. Never see this saphenous vein word before, right? Then what did I say just now about the cholecystectomy thing? Sorry? Yeah, just ignore. Everything else is in syllabus. Okay, like I said, don't see this one already. I never learned before, die. It's always about concepts. It's always about concepts. Okay? Come. 1, 2, 3. Which one? You have a bit for 2. How? Brian, any strong opinions? Destiny? If I were to choose, the most application one is 2. 1 and 3 are very memory based. It's very story based. Very quickly, if you look at question 1, this one, you just need to know what is digested in each part of the digestive system. Correct? You just need to recall what is digested in which part of the... Hello, ladies. Come, let me quickly tell you. Question 1 is about recalling what is digested in each part of the digestive system. Very straightforward. And this one, why must store as starch and glycogen? Because they are insoluble and more compact. You want to be able to store more things in the cell, right? Okay. Removal of the small intestine. Previously, about the removal of the gallbladder, you will approach this question the same way. Just now, remove gallbladder. You think about what the gallbladder does. Oh, now no gallbladder, what happens next? Same thing here. Think about what the small intestine, how the small intestine is relevant for this part of the question. Now, no more small intestine. How? Straightforward. Okay, all right. For question 3, this is a bar graph. I want us to look at this one. Which one is x and y axis? No matter what kind of graph, a graph is still a graph with axes. Which one is x? Try is y. Number of cigarette smoked per person per day is X. Do you agree? Yes, the thing that we control is the X-axis. Well done. And therefore this is the Y-axis. Okay? So even without the bar graph, you can see the trend, right? The more you smoke, higher risk. Okay? Why? Which component of cigarette smoke should we focus on? Tar is one of them. In our syllabus, we have three things in our cigarette smoke. Tar, nicotine, carbon monoxide. Now, nicotine is the one that causes addiction. So that's out. Tar will remain in your airways. It is too big to enter your bloodstream. And hence, we are left with carbon monoxide. For your syllabus, all you need to say is carbon monoxide increases the risk of coronary heart diseases. That's very short question. Two pulmonary conditions that can be caused by smoking. I have to put pulmonary conditions because smoking is this one small little thing that people do that can cause widespread damage throughout the body. Brain cancer, sure. Intestinal cancer, sure. Lots of things. Right? So I wanted to focus on things that can happen to the lungs, the respiratory system itself. So here, what are we thinking of? If one is starting with a B and another starting with an E. I think that's it. Do you remember? Bronchitis. Right? Bronchitis is where the airways become inflamed. Right? Okay. Next one, E. Emphysema. Well done. Emphysema is when Brian, do you know? Remember the name only, Okay, when the alveoli are damaged. Okay, the alveoli are damaged. Go and look it up, right? The revision kits have that. Okay, revision kits have that. Okay, so therefore, let's go. Let's have a look at the heart. Okay, so coronary artery bypass graph. Okay, the coronary arteries are blocked. Okay, coronary arteries are blocked. So therefore, blood cannot flow past the coronary arteries, which means the heart muscles cannot receive enough oxygenated blood. Definitely a bad thing, right? I need the heart to function properly. So I'm going to insert a graft. This graft will allow me to bypass, to skip the blockage. Okay, so if you look on screen over here, I'm just going to highlight to you with this is where the blockage is. Blood cannot flow like that. Can we see that? Blood cannot flow past the blockage. Hence, I insert this graft so that blood can flow this way instead. Okay, so same for here, it's going to be connected to the aorta here, so blood can flow this way instead. Okay, simple as that. A bypass graft allows me to skip a blocked section. So coronary artery bypass graft allows me to skip the blocked coronary arteries. Okay, awesome. So, question is not asking you about the graft or the surgery, but why would there be a blockage? What will cause the blockage? It's about diet. Yes. And so it's excessive consumption. Sorry? So what was that? Sodium. Wow. What? Yes, fats. Yes. Okay. High blood pressure. Hypertension, yes. Okay. So excessive consumption of cholesterol and fats results in these two things doing what? This is the coronary arteries. They start to stick. either is thick, they start to deposit on the walls of the artery. Okay? CA, coronary arteries. If this blockage grows large enough, what would the blockage prevent? Yes, right? Transport. We don't have to elaborate on the effects because question is just asking us to describe the events leading up to the blockage. Okay? But do we know what happened afterwards? If this goes on for long enough, yes, this will result in a heart attack as well. Okay? Okay, finally, the saphenous vein. Like I said, just ignore the word. Simple as that. You can also just ignore the longest vein in the body, resume. That's it. Just need to know the vein. Question is not asking you about saphenous vein. Question is asking you, why is it disadvantageous to use a vein for this procedure? I shared with us just now, this procedure, Shendiao, I'm using a vein to skip what? To bypass what? It's in the question, it's the name, is the name of the surgery is what? I'm bypassing what? Bypassing an artery, thank you. Okay, I'm bypassing an artery, I'm skipping an artery. So if I use a vein to skip an artery, what's going to happen? I use a vein to skip an artery. Think about just now, one of the MCQ, you talk about the structural differences between vein and artery. What? Why will it burst? Yes, possible. Okay, right? The vein has a much thinner wall, and this could result in the future, it could burst then. Right? Okay, so we're looking at differences here. Another one is that the lumen is much wider. Arteries should transport blood at high pressure. The wide lumen will not be able to achieve that. Okay, all right. So we're looking at here we have two possibilities. May burst in the future, or may result in a lower blood pressure to the heart. Okay. And finally, last thing about the vein. Why must it be attached to correct orientation, correct direction? The veins have valves. So a vein has valves and all of them point in the same direction. So if blood is supposed to flow this way, or let me look at this one, right? So blood needs to flow this way. If I attach my flaps this way, blood cannot flow already, right? Okay, fairly straightforward. Must be able to visualize this. Okay, so here it's about function of valves. So we're talking about the valves. Ensure right? Valves ensure unidirectional flow of blood. So you must also think about, hey, then like that, what would happen if the vein is attached incorrectly? Okay, so we add that in. Okay, the last part, I would say this is the part that we are more perplexed with. Can I say that? The pressure graph. Okay, so very quickly, here we are talking about pressure. We're talking about pressure. Just give me a last few minutes, okay? Pressure. And we're looking at semi-lunar valves closing. Right? So we will first think about it. If it's a semi-lunar valve, what two regions does the semi-lunar valve border? Semi-lunar valve is found between which two regions? The left side. Do you remember which two regions of the heart the semi-lunar valve is found between? Shu Ting, do you know this? The semi-lunar valve is found between where and where? The valve found between the atria and ventricles are the bicuspid and the tricuspid. This is the one found between the left ventricle and aorta. Okay, so this one don't copy first. Just look on screen first. This is a suggestion I always like to make when I'm looking at how and when do valves close. This method applies to all valves. You identify which are the two regions the valve is found between first. Next up, you look at this. Oh, close. Valves want to prevent backflow, correct? So if the valve is closed, blood is therefore trying to flow in the correct or wrong direction. If the valve is closed, blood is trying to flow in the correct or wrong direction. Wrong direction. That's why I must close the door, ma. Okay? So therefore, I'll point my arrow in the wrong direction. And follow? Once you are here, then the story is done. P for pressure. The graph is about pressure. So this allows me to find, hey, if I translate this into words, the semilunar valve closes when pressure in the left ventricle is lower than pressure in the aorta. That's it. This method applies to all the valves in the heart. Okay? So very quickly, the last thing for today here, in our worksheets, semilunar valve closes when pressure in LV is smaller than pressure in the aorta. Where is that in the graph? Can I remind us that question is asking for first close? Kathleen, A, B, C, or D? When do the graphs first show me that this happens? That LV pressure is first suddenly switched to become below aortic pressure. Brian, which line do you choose?
B.
B. Brian chooses line B. Thank you. Line B is when the semilunar valve opens. See, come, eyes on screen, please. I highlight for you. This line is aorta line. This line is LV line. Line B is when LV pressure is greater than aortic pressure. So therefore, correct direction. Valve will open. The answer is C when you have the first flip. And this at point C, blue becomes lower than green. At point C is where left ventricular pressure first becomes lower than aortic pressure. That is where the semilunar valve first opens. Okay? All right, so take it slow. This will bring you halfway there. The next thing you need to do is be able to read the graph properly. Okay, so I believe most of us struggled because we couldn't really understand the lines as properly. Take a deep breath, look at the graph properly. Okay, so the answer to this will just be to translate this into words, which you will see in the answer key there. Right, the hard part is looking for that. Okay, can? Right, so we try our best to look through most of the questions you have over here. Some of them, I hope I have managed to help you find out some of the bigger things that you can still have gaps in. Okay, so this kind of short revision things is for you to find out where the gaps you have, and then hopefully this helps you to focus on the topics you should be revising more on when you go back home until your revision that is to come. No, your end of years that is to come. Okay, so as with always, right? Same as what you did with the previous tutor just now. Hope this help you to, if you came in hating bio, I hope you hate it a little bit less. But if not, I hope you are trying to find the light there and appreciating and understanding this beautiful science a little bit more. And as usual, this one, I think he shared with you earlier on already, right? Okay, fairly straightforward. So that was for the chem lesson. If you can help me so much here, please help me to scan this for the bio one as well. Okay, so the QR codes are the same actually. So you can just fill it in twice. Once for the chem lesson, once for the bio lesson here. Can. Hello, can. So I fill in once for this lesson as well. Okay. And yes, we are trying to open up classes everywhere as well, CY. Okay. And yeah, you know what? We have a very much smaller class here. You can take a picture from here. Okay, come, let's go here. Okay, let's go. On the left side, 1, 2. Awesome, thank you. Right, so all the best for your EOYs. If I see you again in future, I will see you again in future. But if not, enjoy the last day of your holidays. Oh, what's my name? Thank you. I took. Yes. Okay, so yes, you I do. And for the rest of us, thanks for learning with me in these 2 hours. Okay, Celine, my tip is to actually be a red blood cell. Think of yourself as a red blood cell, and you think when you're flowing through the circulatory system, oh, on my left side, I pass by the liver. On the right side, I pass by the kidney. What river is this? Is what blood vessel is this? So you think about it this way, you create a story instead of it being a boring from liver to the heart. They're very boring.